Um, so this is really to go over where we are with PARP in the lung cancer space. Um, the title is Strength Through Synergy, Enhancing Treatment with PARP Inhibitors. Uh, now we- So when PARP inhibitors first came into the clinic, the understanding was that these agents would be particularly effective in tumors where there is deficient DNA damage repair. Specifically, there was the expectation that patients with BRCA mutation who are homologous recombinant deficient would be susceptible to PARP inhibition, primarily through the role of PARP in base excision repair, which if you inhibit, will promote formation of double-strand break, which will not be easily repaired because of the compromised homologous recombination repair mechanism in BRCA-deficient tumors. So the earliest exploration of this class of agent was really in patients with BRCA mutations. But subsequently, we've come to understand that PARP plays a lot of role, not just in base excision repair, it also plays a direct role in non-homologous end joining. And this is really by inhibiting the critical enzyme in this process, which is DNA PK. And by doing that, non-homologous end joining leads to poor fidelity DNA repair. So you end up with a lot of DNA, repair DNA with errors that can then lead to cell death. So by inhibiting PARP, you make sure that a lot more DNA damage repair occurs through this mechanism, which will not be high fidelity and then will predispose the cancer cell to death. The other thing that we've come to understand is if for whatever reason you are able to damage DNA, there is always this initial response of PARP enzyme to the site of DNA damage repair. But in order for the repair to occur, you also need the PARP to almost immediately get off of that DNA damage site. So there is the concept of PARP trapping. So you need the catalytic activity of PARP not only to bind to the site, but to also lead to its immediate release from the site to allow the other repair enzymes to bind. So compounds that are very, very potent as catalytic inhibitors are also very, very potent as PARP trappers. And when you trap PARP, then you cannot repair the DNA, and then that becomes persistent and then leads to DNA, cell death. And then finally, because of the role of PARP in serving as a scaffold on which a lot of other DNA damage repair proteins bind and assemble, if you inhibit its catalytic activity, you can also affect different mechanisms outside of homologous recombination and base excision repair. More recently, Dr. DeAndrea from Boston also showed the interaction between PARP and polo theta, which is supposedly very critical for other alternative end joining mechanism. I've gone through this so that we can have a better understanding of why we think we should incorporate PARP inhibitors into tumor types where there is no evidence of BRCA mutation. So what do we know in terms of activity of PARP inhibitors as a single agent? There are many of them in development, and this is a recently published paper in Molecular Cancer Research showing that these agents as single intervention are really not very effective. This is in comparison to temozolomide. But when you combine them with temozolomide, they do much better than single agent alkylating agent. And more importantly, there doesn't seem to be any significant in vivo difference between these compounds, despite the fact that there is a lot of hype about their differences in terms of in vitro activity. This is just a list of currently actively investigated PARP inhibitors. One of them is actually already approved in ovarian cancer, and some are in phase 3 and phase 2 setting. So why are we interested in PARP in lung cancer? So this is work by Lauren Byers from MD Anderson and John Miner from UT Southwestern, where they show that PARP enzyme is actually highly expressed in all variants of lung cancer, but especially so in small cell and large cell neuroendocrine cancers. And talazoparib, which is reportedly the most potent of all the active PARP inhibitors in development, has been moved very rapidly from preclinical into the clinical setting, and initially as a single agent, but I think the strategy is becoming a little bit different following the earlier experience in patients. So this is work by Lauren showing that talazoparib as a single agent in traditional xenografts of small cell, as well as in PDXs of small cell, actually has single agent activity. When this study was being developed, they allowed for patients to be enrolled if they have small cell or germline BRCA mutation in ovarian or breast cancers. And as we can see, these are relatively not so heavily pretreated patients. Majority of them had only the frontline therapies before they went on study. The concern about PARP inhibitors being very toxic has not been borne out by a lot of studies, but we do know that there is always the risk of hematologic toxicity, and the more you have, the greater the catalytic activity, the more likely you are to experience hematologic toxicity. So it's not surprising that in this study, there is really up to 45% rate of grade 1, 2, or 3 hematologic toxicity. How about the efficacy? So within the small cell subgroup, even though there was evidence of this as a single agent in preclinical model, 2 patients, about 10%, achieved partial response. When you compare that to patients with BRCA mutation, whether breast or ovarian, where it was somewhere in the region of 40%, that really speaks to the fact that this may not be a compound that you want to develop as a single agent in this space. So this is an example of one of those small cell lung cancer patients who had dramatic response, as can be seen with this lesion in the liver that almost completely disappeared. And this is really a patient that Lauren Byers treated herself, and she was very kind to share the slide. On our side, we are focused a little more on veliparib, which is an older drug and has been in development at the NCI for almost a decade now, and was shown to have very good PARP inhibitory activity in a phase 0 study. In a phase 1 study that we conducted in Pittsburgh, we showed that we could easily combine this compound with cytotoxic platinum doublet chemotherapy. In the lab, we did not see any significant single agent activity of veliparib in a panel of small cell lung cancer cell lines. But when combined with chemotherapy, in this case cisplatin, there was a dose-dependent potentiation of efficacy, but it's important to recognize that you still need to have sufficient amount of the cytotoxic agent. So this is not a strategy for you to reduce the dose of the cytotoxic. It's more to make the cytotoxic work better at its standard dose. And in the in vivo experiment, not only did the combination of platinum etoposide and veliparib lead to tumor regression, it also delayed tumor regrowth when you observe animals after you've treated for 4 weeks and then you follow them without treatment. Because there is no way to currently predict those who will benefit from platinum, especially small cell, we looked at the correlation between platinum sensitivity of the cell line and the degree of PARP potentiation. And there was actually a modest correlation between sensitivity to platinum and the ability of PARP to reduce the to induce further cytotoxicity. We saw similar but more modest correlation with carboplatin. While the same cells were sensitive to etoposide, there was actually no correlation to etoposide sensitivity, indicating that patients who are sensitive to platinum may be the ones that will benefit from the addition of platinum to chemotherapy. The challenge is how to identify those patients before you treat them. So with that observation, we moved this into the clinic, and this is a study that was conducted through the mechanism of ECOG. A phase 1 study was done showing the safety of the combination regimen, and the randomized phase 2 quickly followed that finished enrollment in July this year with a total of 154 patients enrolled. We're eagerly awaiting the results of this study sometimes in the next 6 months to 9 months. How about in the relapse setting? So this is work by Dr. Kathy Pietanza from Memorial. where they showed single agent activity of temozolomide, but more importantly is the correlation between response and MGMT methylation, indicating that tumors with deficient DNA repair mechanism may be particularly sensitive to temozolomide. So she proceeded to conduct a randomized phase 2 study, which was a multi-center study. And the study enrolled 100 patients. The accrual finished earlier this year, and we expect the result of this to manifest sometimes in the next 3 to 6 months as well. Finally, the Europeans just completed a maintenance study of olaparib in patients who've completed 4 cycles of frontline chemotherapy. It's difficult to know really the final design of the study because it went through so many amendments, and depending on which version you read, it kept changing. But whatever it is, the study completed accrual, and they were looking at both BID as well as TID schedule of olaparib. So it's a 4-arm study, but the total dose of olaparib is the same in both arms. It's just a matter of whether you give it BID or TID. So hopefully we'll get the result of this study also next year or the year after, because it's a much larger study and looking at maintenance. How about non-small cell? So the story is not just with small cell. People have also looked at the role of PARP as a potentiating factor in platinum doublet chemotherapy. So this is a study led by Dr. Ramalingam. It's a randomized phase 2 study combining veliparib to carboplatin paclitaxel, and that was compared to standard chemotherapy. The study enrolled approximately 160 patients globally, and the 2 groups were well balanced. And in terms of adverse events, which is what we worry about, as you can see, there's a slight increase in leukopenia in patients who received veliparib along with chemotherapy, but beyond that, there was really no signal of increased toxicity when you add veliparib to platinum doublet in this setting. And this is the result based on RECIST criteria. As you can see here, the response rate in both groups is actually comparable, 28% to 31%. What is remarkable, though, is the duration of response for patients receiving veliparib along with platinum chemotherapy, where it's more than doubled, which speaks to the fact that you cannot use this to dose reduce your cytotoxic agent. It's more about further potentiating the activity. And you can see that here, that while the proportion of patients responding is more or less the same, remember this is 2-to-1 randomization, the depth of response is deeper with the triplet chemotherapy. Did this translate into survival advantage in the overall patient setting? No, there was really no PFS or OS advantage. However, in subset analysis, which was pre-specified, there was a trend in patients with squamous histology that the addition of veliparib may lead to improved survival outcome. Whereas when you look at patients with non-squamous histology, there was really no difference between the 2 groups. So this study has now moved forward into a randomized phase 3 study, which is actually doing very well ex-US, and hopefully should finish accrual in the next year or so. So in conclusion, PARP enzyme is a validated target of therapy in lung cancer. Veliparib, as I've shown, as well as olaparib, are currently in frontline setting therapy and maintenance therapy setting for small cell. Combination of veliparib with platinum doublet is very promising in squamous variant of non-small cell. But I think identifying specific predictive biomarkers will be very, very important in integrating this promising compound into the management of lung cancer patients. Thank you.
Somebody was. Is there a hypothesis for the adeno or the non-squam versus squam efficacy?
Yeah, so there is some hypothesis behind it, and one of the hypotheses actually is similar to what we're talking with immunotherapy. So the squamous patients tend to be heavy smokers. You have a lot more DNA damage in those patients, and it seems like those patients seem to be particularly sensitive to PARP inhibition. That's just hand waving, but that is what people are thinking about. So.
And Taofeek, if I can add to that, in the same study, when we looked at outcomes based on smoking status, patients with smoking-related cancers, regardless of histology, did significantly better with the addition of PARP inhibition to chemotherapy, and the hazard ratio there was 0.36. So it trumped the histology effect. And the company is doing a trial in patients with smoking-related lung cancers, also looking at similar combination. But my question was, can you comment on what's the latest on PARP trapping and how it varies from one drug to the other?
So 3 years ago, the NCI group came and said it's not the catalytic activity that matters, it's the PARP trapping, and then everybody went in that direction. So the paper that I just showed actually very beautifully showed that PARP trapping, while it is important, the catalytic activity is equally important, and that it was thought that the PARP trapping is an allosteric interaction between PARP and the DNA, and they show that it's actually the catalytic activity that mediates the trapping. So if you have very good catalytic activity, you're going to have very strong PARP trapping effect as well. But they showed that the PARP trapping effect did not translate into in vivo efficacy. So it may be mechanistically relevant for identifying promising compounds, but whether or not it will translate into clinical difference remains to be seen.